Hey, welcome back to Following Signs, the podcast that takes you on a journey throughout the breakthroughs, redefining how we understand and interact with the world. I'm your host, Rutu Parl, and today we're diving into a topic that lies at the very heart of modern cancer care: biomarkers. Cancer is not a single disease. It's an umbrella for over 200 different diseases, each with its unique characteristics, behaviors, and genetic signatures. Historically, we approach cancer with a quote unquote one size fits all strategy, relying on surgery, chemotherapy, or radiation. But in recent decades, the discovery of biomarkers has shattered this one dimensional approach, allowing us to understand cancer at its molecular level. So, in this episode, I'm going to be breaking down the concept of biomarkers and their evolving role in cancer care. Then I'm going to explore how genes and mutations act as a foundation for biomarkers discovery, and then go into the cutting edge technologies that enable us to find these molecular cues before finally exploring how biomarkers are reshaping cancer therapy with precision medicine and immunotherapy, while also going over the challenges that researchers are facing and what the future might hold. Now, today's discussion won't just skim the surface. We're going to go deep into the molecular biology and the tools and the real world impact of biomarkers. So, whether you're a seasoned researcher, a curious student, or someone just impacted by cancer, this episode has something for you. Now, let's start with the fundamentals. What exactly is a biomarker? The term biomarker is short for biological marker, and it refers to any measurable characteristic in the body that indicates a biological state, condition, or process. Biomarkers can be simple as blood pressure reading or as complex as genetic mutation found in tumor cells. In cancer care, biomarkers are particularly powerful. They help us, first, diagnose cancer. Biomarkers can detect the presence of a tumor, sometimes even before symptoms even appear. Second, they help us predict outcomes. Prognostic biomarkers can determine how aggressive a cancer might be or how likely it is to spread. And finally, three, it helps guide treatment. Predictive biomarkers show us which therapies are most likely to be effective for a specific patient. Now, the concept of biomarkers isn't new. In fact, doctors have been using basic biomarkers for centuries, long before they knew what they were. For example, fever was recognized as a biomarker of infection, and sugar in urine pointed to diabetes. But the era of cancer biomarkers began in the mid 20th century. One of the first milestones was the discovery of CEA, which is carcinoembryonic antigen, in 1965 by Dr. Phil Gold and Dr. Samuel Friedman. Now, CEA is a protein found at higher levels in the blood of patients with colorectal cancer. Although not perfect, since elevated CEA levels can also occur in smokers or those with benign conditions, it marked the beginning of using molecular markers for cancer detection and monitoring. Now, let's go into exploring the different types of biomarkers, which I kind of already did mention a little bit earlier, but now I'm going to go into a little more detail. So, let's start with diagnostic biomarkers. Now, these biomarkers detect a cancer's presence in the body. A good example is prostate specific antigen, or PSA, which is a protein produced by prostate cells. Now, elevated PSA levels in the blood can indicate prostate cancer or benign conditions like prostatitis. Now, although controversial due to false positives, PSA screening has saved countless lives by detecting prostate cancer early. Then, let's talk about prognostic biomarkers. A prime example is the Oncotype DX test used in breast cancer. Now, this test analyzes the expression of 21 genes in a tumor and assigns a recurrence score. A high score suggests a greater risk of the cancer returning, guiding decisions about whether chemotherapy is even necessary in the first place. Finally, let's talk about predictive biomarkers. Predictive biomarkers are revolutionizing cancer treatment. One standout example is HER2, or human epidermal growth factor receptor 2. Now, HER2 is a protein that promotes cell growth. Now, about 20% of breast cancers have an overexpression of the HER2 gene, making the tumors aggressive. However, these tumors respond exceptionally well to HER2 targeted genes like herceptin. Now, let's take a closer look at AFP, a biomarker for liver cancer. AFP is a protein normally produced by the liver and yolk sac during fetal development. In adults, high AFP levels can signal hepatocellular carcinoma, or HCC, which is a common type of liver cancer. Now, AFP isn't just a diagnostic tool, it's also used to monitor treatment response and recurrence. But there's nuance here. Not all liver cancer patients have elevated AFP levels. In fact, some cases of HCC are AFP negative, prompting researchers to look for additional biomarkers like DCP to improve detection accuracy. Now, today, researchers often use a panel of biomarkers instead of just relying on one single marker. For example, the Cancer-Seq test combines several biomarkers and DNA mutations to detect multiple types of cancer with a single blood test. This approach significantly improves sensitivity and specificity, reducing false positives and negatives. By understanding biomarkers as molecular cues, we're better equipped to appreciate how they connect to cancer's genetic underpinnings, which we'll explore in the next segment. Now, to truly grasp the power of biomarkers, we need to understand their genetic roots. At its core, cancer is a disease of the genome. Our DNA, composed of genes, act as the instructional manual for building and maintaining our bodies. When these conditions, or rather, when these instructions are altered through mutations, deletions, or duplications, cells can lose control, and then they become cancerous. Now, genes can be broadly categorized into two groups when it comes to cancer. First, oncogenes, and second, tumor suppressor genes. Let's go into more about oncogenes, because they're like a car's gas pedal in a sense. Because when functioning normally, oncogenes regulate cell growth, but when mutated, they get stuck, driving uncontrolled cell division. Some examples include HER2, which is the overexpression of HER2 leads to aggressive breast cancer, and KRAS, where mutations in KRAS, common in colorectal and lung cancers, make cells resistant to certain treatments. And second, of course, there's tumor suppressor genes. These act as the brakes on cell growth. When mutated, the brakes fail. Examples include stuff like TPF3, which is often called the guardian of the genome. Now, TPF3 mutations are often found in over 50% of all cancers, leading to unchecked cell division. And of course, there's BRCA1 and BRCA2. Mutations in these genes impair DNA repair, significantly increasing the risk of breast and ovarian cancers. Now that we understand what biomarkers really are and how they kind of relate to genes and cancer, let's dive into the fascinating technologies that drive the discovery and application of these biomarkers today. In the past, identifying biomarkers was a time-consuming and often imprecise process, but with advent of powerful new tools, researchers are now able to pinpoint genetic alterations and protein changes with greater precision and efficiency. Now, one of the most revolutionary technologies in biomarker discovery is next generation sequencing. Now, in simple terms, NGS allows us to read the entire genetic code of a tumor in a fraction of the time it would have taken using older methods like Sanger sequencing. Now, this technology can sequence millions of DNA strands simultaneously, offering a comprehensive view of the genetic makeup of both normal and cancerous cells. Now, NGS works by fragmenting DNA into small pieces, attaching adapters to the ends, and then amplifying and sequencing these fragments. These sequences are then compared against reference genomes to identify mutations, insertions, deletions, and other alterations. This allows the researchers to discover somatic mutations, those that occur in the tumor cells and are not inherited, and of course, germline mutations, which are inherited and many may predispose individuals to cancer. Now, one of the most comprehensive resources to emerge from the NGS technology is the Cancer Genome Atlas, or TCGA. Now, this is a project led by the National Cancer Institute, or NCI, and the National Human Genome Research Institute. Now, this project has mapped the genomic changes in over 30 different types of cancer, providing a blueprint of genetic alterations and uncovering hundreds of potential biomarkers. For example, in lung adenocarcinoma, TCGA identified mutations in the ECFR gene that could be targeted by specific drugs. Now, another cutting edge technology making waves is the liquid biopsy. This non invasive test analyzes blood samples to detect DNA, RNA, and proteins released by tumors into the bloodstream. It's an ideal method for tracking the genetic evolution of cancer without requiring invasive tissue biopsies. Liquid biopsies can also be used to identify circulating tumor DNA, which carries mutations specific to the tumor, providing real time insights into the cancer's genetic makeup. Now, one of the major promises of liquid biopsy is early detection. For example, Guardant 360, a commercially available liquid biopsy test, can detect genetic mutations in patients with advanced solid tumors, helping to guide treatment decisions. What's truly innovative about liquid biopsies is that it enables the monitoring of treatment response over time. Instead of waiting for clinical symptoms or imaging changes, a simple blood draw can provide valuable data about the tumor's genetic evolution, including the emergence of drug resistant mutations. However, liquid biopsies is not without its challenges. For instance, ctDNA levels may be low in early stage cancers, reducing sensitivity. Now, additionally, the technology is also still being refined for routine clinical use, but the potential for non invasive real time monitoring of cancer progression is still a game changer. Now, another powerful tool in the biomarker discovery arsenal is single cell RNA sequencing. Now, traditional sequencing technologies analyze bulk tissue samples, which represent the average genetic profile of all cells in the sample. However, tumors are heterogeneous, meaning they consist of a mixture of cells with different genetic alterations. This heterogeneity makes it difficult to identify biomarkers that represent the diversity within a tumor. Now, single cell RNA sequencing isolates individual cells and sequences their RNA, providing a detailed picture of gene expression at the cellular level. This allows researchers to identify rare cell populations within a tumor that may be driving cancer progression or resistance to treatment. For example, in glioblastoma, a highly aggressive brain cancer, researchers have used scRNA sequencing techniques to identify rare cancer stem cells that are resistant to traditional treatments. By targeting these cells, scientists hope to develop therapies that can effectively treat glioblastoma. Now, single cell sequencing is also being used to study the tumor microenvironment, this ecosystem of non cancerous cells, blood vessels, and immune systems, or rather immune cells, surrounding a tumor. The TME plays a critical role in cancer progression, and by understanding it at the single cell level, we can identify novel biomarkers and therapeutic targets. Now that we've looked and explored the cutting edge technologies used to identify cancer biomarkers, let's take a closer look about how these biomarkers are being used in cancer therapy specifically. Now, cancer therapy is becoming a lot more personalized thanks to advances in precision medicine. Now, in traditional treatments, patients are grouped together based on their cancer type and treated with the same drug. But now, precision medicine allows us to tailor treatments to individual patients based on their tumor's genetic profile. Now, one of the most significant advancements in cancer treatment is targeted therapy, which uses drugs or other substances to specifically attack cancer cells without harming normal cells. Biomarkers play a pivotal role in identifying which patients will benefit from these therapies specifically. Now, let's consider an example. Considering epidermal growth factor receptor, which is a protein found on the surface of cells that helps regulate growth. Now, in some non small cell lung cancers, mutations in the EGFR gene cause it to become overly active, leading to uncontrolled cell division. Patients with EGFR mutations can be treated with targeted inhibitors such as Tarceva and Iressa, which block the mutated receptor and stop the cancer's growth. These drugs are incredibly effective, but only for those with EGFR mutation. This is where biomarker testing becomes critical. It helps doctors identify the patients who will benefit from these therapies specifically. Now let's talk about immunotherapy, which harnesses the body's immune system to fight cancer, and it has been another revolutionary treatment. However, not all patients respond to immunotherapy, so understanding which tumors are likely to be sensitive to these treatments are crucial. One key biomarker in immunotherapy is the programmed death ligand 1 protein. Now, this PDL1 is often expressed on tumor cells to evade detection by the immune system. Drugs like Keytruda and Opdivo block the PD1-PDL1 interaction, allowing the immune system to recognize and attack the tumor. However, these drugs only work in tumors that express high levels of PDL1. Therefore, testing for PDL1 expression is essential before starting treatment. Now, another critical immune-related biomarker is tumor mutational burden. Now, TMB refers to the number of mutations present in a tumor's genome. Tumors with high TMB are more likely to produce abnormal proteins that the immune system can recognize as foreign, making them more susceptible to immunotherapy specifically. Now, while biomarkers like PDL1 and TMB have shown promise, immunotherapy still faces challenges. For instance, not all tumors with high TMB respond to treatment, and the role of PDL1 is not always straightforward. Researchers are actively exploring additional biomarkers, including those related to the tumor microenvironment and immune cell infiltration, to refine patient selection for immunotherapy. So, what does the future hold for biomarkers in cancer research? The rapid advancements we've seen over the past decade are just the beginning. As technologies improve and understanding of cancer deepens, we can expect biomarkers to play an even more integral role in early detection, diagnosis, and treatment. Now, one of the most exciting developments on the horizon is the integration of artificial intelligence, or AI, and machine learning, or ML, into biomarker discovery. AI and ML algorithms are increasingly being used to analyze large datasets, identify patterns, and predict the presence of biomarkers. These technologies have the potential to significantly accelerate the pace of biomarker discovery by analyzing vast amounts of genetic, proteomic, and clinical data. For example, AI can be used to analyze the genomic data from NGS, searching for novel mutations that may be overlooked by traditional analysis methods. AI can also help predict how a tumor will respond to a specific treatment based on its genetic makeup, providing doctors with personalized treatment options. Now, in the coming years, liquid biopsy technology will continue to evolve, enabling even earlier cancer detection. Researchers are working to improve the sensitivity of liquid biopsy tests, making them suitable for screening asymptomatic individuals and detecting cancers at their earliest and most treatable stages. Some companies are already even developing liquid biopsy tests that can detect cancers in their early stages long before symptoms appear. Now, these tests could one day become routine screening tools, similar to mammograms for breast cancer or colonoscopies for colon cancer. Now, the future of cancer biomarker discovery is also heading towards multi-omic approaches, where data from multiple layers of biological information, such as genomics, proteomics, and metabolomics, are combined to identify biomarkers. This integrated approach will allow for a more comprehensive understanding of cancer and its biomarkers, ultimately leading to a more effective and personalized treatment. Now, as we've explored today, the realm of cancer biomarkers is vast and continuously evolving. From their role in early detection to their impact on personalized treatments, biomarkers are transforming the way we understand and fight cancer. The combination of cutting edge technologies such as next generation sequencing, liquid biopsies, and single cell RNA sequencing has already made a monumental difference in advancing cancer research, and we're just scratching the surface of their full potential. These advancements are not only improving how we detect cancer, but also revolutionizing how we treat it. Biomarkers are now guiding the way for targeted therapies and immunotherapies, allowing us to personalize treatments like never before, sparing patients from unnecessary side effects and, of course, improving outcomes. Now, looking forward, biomarkers not only hold the potential to detect cancer earlier, but they can also help us better understand why certain treatments work for some patients and not for others, ultimately leading to more tailored, effective cancer care. Now, as I wrap up today's episode, I just want to thank you all for joining me again on this episode on Following Science. I really love talking to you guys, and so every time you reach out, it just makes my heart happy. So please keep reaching out with your questions or topics you want me to explore. And of course, thank you for joining me again today as we explore the cutting edge world of cancer biomarkers. It's clear that the future of cancer research is more hopeful than ever, and with continued innovation, we'll keep pushing the boundaries of what's possible. Stay tuned for more in-depth discussions and updates on the latest scientific advancements on following science. Until next time, stay curious, stay informed, and keep following science. This is your host, Ruto Parle, signing off.